Morning student, today we will study about the gene ecology. Ecotype. In gene ecology, one or more local population of single species are studied to know the species genetically determined, tolerant to environmental condition. In course of time, individual of a species with genes suited to a local environment get selected by natural selection and constitute a local population. In a given climatic area, all the local population develop similar environmental tolerance and constitute an ecotype or an ecological race. This was recognized for the first time by Tursson of Sweden in 1922. His experiment revealed that a taxonomic species is not a single ecological unit but is composed of numerous races which exhibit inheritance difference in physiology and morphology. These differences were usually correlated with specific type of habitat. He used the word ecotype for a group of local population of a species from the same type of environment and showing similar inherited morphological character. According to Turshan, local population were ecologically adapted genetic group within a series. The basic proportion of Turshan's gene ecology may be summarized as wide-ranging plant species shows spatial variation in the morphological and physiological character. Much of these intraspecific variations can be correlated with the habitat difference to the extent that the ecological correlated variation is not simply due to the plastic response to environment. It is attributed to the action of natural selection in molding locally adapted population for the pool of genetically variation available to the species as a whole. In his exemplified paper, it is shown that ecologically correlated interpopulation variation was commonly genetically based rather than dependent upon the direct modification of the individual. This conclusion led to the concept of ecotype. The most significant statement made by him was the mass of genetically distinct form which make up the linear species do not distribute themselves indiscriminately over an area comprising different type of localities, but on the contrary are found in the nature to be grouped into different type, each confined to a definite habit. Further, these ecotypes do not originate through sporadic variations preserved by the chance isolation. They are on the contrary to be considered as the product arising through sorting and controlling effect of the habitat factor upon the heterogeneous species population. Since the differentiation into the ecotype result from discrimination selection offered by unlike habitat, it follows that in general, the wider the ecological range of the species, the more numerous are its ecotype. Wider ecological range of a species may be due to plasticity of genome or to multiple allele. Since ecotypes are interfertile, they are never pure. In a region where the range of two ecotypes overlap, they exchange genes freely. Even through outside this region of contact, the two types exist unaltered because of continuous pressure of environmental selection. New ecotype may be formed due to hybridization, slow accumulation of numerous small gene change which are individually insignificant may also result in the development of new ecotype. A third manner of the origin of ecotype is the chromosomal changes. For example, polyploid rarely exhibit the same ecological amplitude. Cultivation of a species result in the origin of several ecotypes. This is because under cultivation there is relatively little competitive selection, which is in nature would promptly eliminate many innovation. Ecological significance of ecotype. The degree of ecological amplitude has an important bearing on the possibilities of natural or artificial extension of a species range. Man has been able to extend the range of the economic plant so widely only because he has produced new ecotype by adapting cultivation practice which compensate for time climatic diversity and at the same time keep the plant free of serious competition for natural vegetation. Thank you.